Welcome back to another episode of the BioHub. Today, I'm joined by John Mumm, the CEO of Deka Biosciences, a company breaking new ground in cytokine therapies for cancer and inflammatory diseases. John is a true veteran of biotech with over 30 years of experience in immunotherapy research. He spent his career working to unlock the potential of IL-10 and cytokines, tackling one of drug development's biggest challenges. After leadership roles at MedImmune and ARMO BioSciences, he's founded Deka to bring a fresh approach to cytokine-based treatments. Today, we dive into John's journey as a three-time biotech founder, the fast-moving progress at Deka, and why cytokines have been tough to crack. Plus, we get into his mindset, motivation, and what it takes to build a company that lasts. So, let's get into it, John. First of all, welcome. Glad to have you.
Thanks, Max. Appreciate it.
So, John, another startup. What just took us through your journey so far in biotech, and then also what has then led you to found Deka?
Yeah. So I think I can characterize my journey in biotech as resilience and luck. So first, grew up in a super small town, didn't have a lot of options. There was a wandering, itinerant kind of speaker that told us once in a senior year kind of assembly that our journey, our vocations, our jobs hadn't been invented yet. And for me, that was kind of a clarion call that, to open my eyes. When I went to college, it's, what else is out there beyond what I may have grown up with? And, sure enough, that was the first luck. The second luck was getting into a super small college that had what I think is maybe the only biotechnology baccalaureate degree ever. And part of that degree is that in our first few months, every Friday we had a freshman dinner seminar. And my second freshman dinner seminar was actually the speaker was one of the founders of Core Therapeutics. And he told this story about trying to develop drugs for people to make their lives better. And 8 members of my family had died from cancer. So that really kind of resonated with me. And he talked about the fact that he was in academia and he couldn't kind of, get the people together to really think about making a drug. And then he went to pharma and he couldn't get people in a fast enough time frame to figure out how to make a drug. And this is kind of small molecules for cardiovascular disease. And then this is 30 something years ago, kind of the nascency of the biotechnology industry. And then he talked about founding Core Therapeutics and how they quickly were able to take all of this accrued knowledge and make a therapeutic for patients suffering from cardiovascular disease. And that just resonated. That was it. Like, that was my path. Now, in undergrad, I had to work full time. So I had a job in a biotechnology pharmaceutical executive recruiting firm. And I was on the research side. And I saw what the executives, I saw what their path was. It was, undergraduate, graduate, postdoc, whether it was or a residency, right? So whether it's a PhD or an MD. And keep in mind, no one in my family had ever gone to college. So for me, this contemporaneous, being in school for science and really loving science and then working full time and seeing what the career path was, it was kind of a no-brainer for me what to do. And so I told my girlfriend at the time, who is now my wife for 30 years, I was like, I'm going to get a PhD so I can be a CEO. And she said, you've lost your mind. People don't get PhDs to become CEOs. At the time, they didn't, right? Because biotech wasn't a thing. So, I had this job, making pretty good money, but I had a career requirement to actually do an internship at a biotechnology company, which I did. And I fell in love with benchtop research, with benchtop science. And it was a biochemistry group. It was Energen. Energen was the name of the company. They were trying to basically build MHC class 2 molecules that with an appropriate peptide that would deliver an energic and attenuating signal to autoreactive T cells, right? So from the very beginning, I was playing at the interface of how the immune system speaks to the body in a disease state, in autoimmune disease. And then when I went to Stanford, the flip side was, well, if the immune system can inappropriately destroy self-tissue, can you turn it on to target cancerous tissue? So I did my master's, worked with Mark Davis at Stanford, and I worked with Peter Lee. He was my benchmate. And I built class 1 tetramers. So John Altman had made this tetramer technology. I took it up. I was a kind of a protein biochemist. I could fold proteins pretty well. And so I made all of these class 1 tetramers with putative tumor antigens that no one else had been able to make. And then Mario Schroeder down in Lee Herzenberg's lab in the basement of the Beckman Center at Stanford had a 9-color flow that he had basically built from scratch. And so I made the tetramers, and Peter he's a hematologist, he got the blood, and we go down to Mario and we ask the question, do cancer patients have tumor reactive T cells, CD8s? And if so, what is their phenotype? And what we found is that all cancer patients that we tested had tumor antigen reactive T cells that were of a memory phenotype. So what that means is the whole immunological train had worked in those patients, right? The antigen presentation and clonal expansion of the CD4s and of the CD8s and activation and trafficking out of the lymph nodes back to the tumor, that had worked. What it then meant was that it had failed at the tumor. So when I went to do my PhD with Liz Grimm, so Liz Grimm worked with Steve Rosenberg to put IL-2 into patients. So, IL-2 was a big thing in that lab. I asked the questions of the cytokines that are present in tumors, what feeds back to shut down an active immune response, CD8s in particular. And so Jack Roth, the surgeon, Liz's husband, we went and got tissue from him, melanoma tissue. We grew out the tumor cells, we grew out the T cell clones, and then I put cytokines onto the T cell clones to see what would shut down the response to the autologous tumor. And what I found was the opposite of what I should have found, which is the more IL-10 I added, the more activated the CD8s were. Well, I thought I did it backwards. I thought, you're an idiot, do it again, right? You really got to get it together, John. So did it again, same thing was true. So what it meant was, what I had inadvertently found is if you have human tissue, human CD8 T cells with their cognate autologous tumor, the more IL-10 you add, the more capable they are of killing the tumor cells. Now keep in mind, you cannot carry T cells in a dish without IL-2. So I was already combining IL-2 and IL-10 in a dish. And that resulted in profound destruction of tumor cells. So took that story forward through my thesis. I then, because I'm working in cytokines, managed to get a job with Martin Oft at DNAX, where IL-10 was found. And I actually went to every single PI that had ever worked in IL-10. So Kevin Moore, he discovered it. Kevin was working with Peter Van Blessler at Arresto. So that's the first time I met Peter Van Blessler, who I would then work with in the future. Kevin said, this is really cool data, John, but I'm not working on IL-10 here. I then went to Maureen Howard, that was at Chemocentrics with Tom Shaw. Maureen and I had worked together at Anergen, and Maureen said, this is really cool. And she had been at DNAX. She said, this is super cool data, John, but I'm not working on IL-10 here. I'm like, all right. So then I'm like, I'm going to drop the IL-10 thing. I'm just going to get a job at DNAX. So I managed to convince the DNAX crew and Martin that I could work on cytokines. And so I actually started working on P19, which is the subunit of IL-23, right? That's in the inflammatory circle. So June 5th, 2005, I did a Western blot for P19. June 6th, 2005, I started growing cells to put into mice to do my first PEG-10 study. Because IL-10 was discovered at DNAX, Schering Plough, they literally had pegylated IL-10 sitting in the freezer and nobody was using it. So what then happened is, for the first year of my postdoc, and I only postdoced for a year, I pushed the P19 story forward in cancer and in inflammation, and I pushed the PEG-10 story forward to treat mice, because I'd already figured out everything in a dish, to treat mice with PEG-10. And what I found is PEG-10 is a beautiful molecule to cure tumors in mouse, and P19 and IL-10 cross-revolve around each other. So if you have a circuit of IL-23 and you give IL-10, you shut it down, right? So when you think forward to Deka, I have IL-10 for oncology and I have IL-10 for anti-inflammatory, right? So just, again, purely by luck, I've constantly been on a path that has this therapeutic capacity with an evolution of knowledge to be able to use usefully for people. All right, so PEG-10, postdoc for a year, get hired as a scientist. I think of the, couple hundred postdocs that went through DNAX, a very small percentage got hired as scientists. It's a pretty high bar. It was a phenomenal place to work. was lucky enough to get hired as a scientist. And for the next 4 years, we pushed the PEG-10 story forward, feed forward or roll forward. We're about to put everything together for an IND, and Schering-Plough and Merck do their reverse merger. And so I'm sitting there. Now, keep in mind, the Merck back then was not the Merck of today. So what Merck was back then was a bunch of small molecule folks. And so I'm sitting there thinking, not only am I saying that I'm not working on an antibody, I'm working on a small cytokine where everyone says it's a suppressor, and I'm saying that it's a stimulant. There is no way that the Merck folks are going to look at me with anything other than complete disbelief and a clear understanding that I'm insane. So, I've been in small biotechs. When I was down in Texas at MD Anderson, I worked in small biotechs. Like, I've always had one foot in the biotech world. And I went to Martin and I said, look, we need to start a company to out-license PEG-10 because they're not going to take it forward. And Martin was like, you're nuts. I'm like, no, I can put together a little bit of money. Like, let's do it, right? And Martin's like, all right, fine. Now, right at that time, because the reverse merger, Rob Castelline, who was the site head, needed to downsize, okay? Now, this is one of the things that, I always tell my kids, there are events that happen in your life that at the time seem terrible, but in retrospect are amazing. This is one of those. So Rob comes up to me and I'm working in a lab and he goes, John, I hear that you want to start a company. I'm like, absolutely, right? He's like, okay, right? He has to downsize. So in Rob's mind, he's like, check, I've got one of my scientists that is ready to leave. I'm going to give him a package, right? I'm going to help him out. It's all good. And Rob did an amazing thing for both Martin and I. So we were downsized. We had 3 months to actually get our manuscript ticked and tied, and then 6 months severance. So it was brilliant. Like, we had all the time in the world to actually kind of get our act together. So we did. We left. We got a term sheet from MedImmune for acquisition. That wasn't quite enough for Merck to let it go. And keep in mind, PEG-10 and anti-PD-1 were both on the out-licensing docket at Merck, right? Martin and I managed to get the PEG-10 out. Andrea Fanelsis and Viva Olive, who had made the anti-PD-1, were just about to pull it out when Roger Perlmutter said, no more out-licensing. And that is what became Merck's anti-PD-1. So, like, right there, right? All of that science was happening at the same time. So Martin and I, so Rob introduced us to Peter Van Blessler. Peter Van Blessler takes the term sheet from Medi, takes an understanding of all of the data that Schering-Plough, DNAX, Merck had accrued on 10 and PEG-10. And he goes, all right, boys, we're going to start a company. And I was like, hot damn, hallelujah, let's get it done. Because that's all I ever want to do. I love starting companies. I love growing companies. And so, I worked with Peter for the next 4 years. We had ascending term sheets every year. I learned CMC, which I knew nothing about to start with. I thought it was critical micelle concentration until I looked it up. And Peter is a CMC expert. He's a brilliant scientist, but a CMC expert. And so I managed to learn, a huge amount in that period of time. We did 28 at scale, campaigns, GMP campaigns. That's, if you're going to learn something, you gotta do it. And so kind of with Deka, it was the accrual of all of that information of how to make cytokines work better and how to actually make them kind of able to be manufactured. So that was really, we got ascending term sheets. We got our first $1 billion term sheet. And I thought, okay, I think I've learned enough. My daughter had just gotten into Princeton. I wanted to be closer to her. So I reached out to MedImmune and got a job at MedImmune with a bunch of folks that I knew. And at MedImmune, I saw the unfortunate demise of all of the co-stimulators, right? So the 4-1BBs and OX40s, et cetera. And I realized, what we were trying to drug with those molecules is what cytokines do naturally in a soluble state. And I thought, it's time to actually start a company to solve the challenges with cytokines. And the challenges with cytokines is, as I mentioned, they're not designed to be drugs. They're super small, and they need to be combined and enriched in affected tissue. So because of the evolution, right? Keep in mind, I was in biotech from the beginning. The barriers to entry 30 years ago were profound compared to now. So Pavel and I started Deka. I put in about, $500,000. And I said, let's ask the question if it's possible to structurally combine IL-2 and IL-10 in a targeting vector to actually drive the biology that we need that we need to drive? And is it easy enough to manufacture that with a very small lab, you can make it and it's stable enough that you can actually keep it in the fridge and work with it? And the answer was yes. So that piece, those couple sentences I just said, when I talk about luck, it was pure unadulterated luck that the combination of these molecules together, A, works, B, is expressed, and C, is unbelievably stable. We're stable for at least 2 years just sitting in the fridge, right? That's unheard of for this kind of what I call Franken molecules, right? Because there's 3 different domains. So, luck is important, but also the other piece is when you start a company, and I think the reason that we've moved as quickly as we have is that we started with a very explicit ask, which is, can they be made? If they can be made, are they stable enough to use over a period of time? And if those 2 things are true, if the CMC, if you will, is sorted first, then does it actually elicit the biology? And so I have a story for you. When I was doing my graduate work, Liz Grimm would always tell me, John, you have to form a hypothesis. And I would say, Liz, I'm too stupid. I don't know enough to form a hypothesis. And what I mean is, forming a hypothesis has a particular kind of arrogant bias in the sense that you know so much, you can actually predict how a system is going to work or how a system will be perturbed with a given intervention. Okay. Now, as a young scientist, I don't know that anybody has kind of all that knowledge. Maybe you read 8,000 manuscripts and, or something like that, and you've got a super big brain. I was never that person, right? I mean, I love to read, but I don't read every manuscript ever. And I just looked at it realistically. I said, Liz, I don't know. I can't form a hypothesis. I'll set up a system. I'll control my inputs, and then I'll look to see how the system is perturbed. I'll do that. So with Deka, I made a couple hypotheses, right? So that IL-2 is toxic, predicated on the induction of cytokine release syndrome, and IL-10 as an anti-inflammatory controls it. No one had ever shown that IL-10 could control other cytokines' induction of inflammation, right? IL-10's anti-inflammatory function is on monocytes responding to bacterial products. Okay, that's the first thing. The second thing is that, at ARMO, we found in our cancer patients that when you treat them with PEG-10, you block Tregs. Well, Tregs are a liability of IL-2. We published the fact that if you combine IL-2 and IL-10 in a dish, that if you give 10 to 100-fold more IL-10, you could block IL-2's induction of Tregs. So the second hypothesis was that if I combine them at a molar equivalency, right, in the same molecule, I'll shut down the Treg liability. And then the third was targeting to the tumor would enhance function. That was kind of a no-brainer, but you have to do it. And so we proved all of those in a very short period of time because we had very explicit ask, and then we had a very targeted answer, right? Up or down. And those were my hypotheses. And when I proved them, I called up Liz and I said, hey, I made my first hypothesis. And she said, John, you now deserve your PhD, right? And that's, it's important. These pieces to an evolution of an intellect, to accrue enough information to be willing, right? Because as scientists, you go through a thesis defense, they're trying to shoot you down, right, left, and center. You do your postdoc, they're trying to shoot you down. You put out your first manuscript, they're trying to shoot you down. It's always a process of kind of resolve and resilience and improving your own intellectual capacity. So that was really this initial test. Can we do this? And the answer was yes. And then it was how fast, right? Because you never get back time. How fast can we do the rest of it? And so, the milestones that we've hit is within basically 2 years, we'd established the platform. We then worked with Chris Yen, who's our corporate counsel. He was a former U.S. patent examiner. So he kind of knew the inner workings of the USPTO. So we negotiated with the patent office that every single sequence of what we call a diakine, the combined and target cytokines, was its own composition of matter, its own patent, right? So we have 21 granted patents because we basically understand the system and the business side of things, right? So we now have the ability to essentially confirm to any pharma whatever molecule they want us to build, that is unentangled. It's disentangled from an intellectual property perspective to any other molecule. It's theirs, right? And that's important when you build a platform, right? So, we established it, we secured the IP, and we actually did a tox study. So I paid for an initial tox study to prove, right, that in fact, when you couple 2 and 10 together, you take away the massive toxicological profile of IL-2. And you take away the induction of Tregs. So we proved that in monkeys first. That was before we got our Series A. So I think that, the other piece is when it's your own money at play, and I've continued to put money in, but when it's your own money at play, there's an expediency to ensuring that you're not wasteful and that the questions you're asking are actually relevant to the answer of the therapeutic. There's a lot of questions you can ask. You can get lost, like I said from the beginning, this whole, cytokine does this and cytokine does that and all that craziness depending on the cell. You can get lost in the noise of what you're trying to do. But ultimately, we have a clear understanding. We're making drugs, right? And I told the team for about the first 4 years, I was like, we don't do science. We do drug development. Those are very different things, right? Now, because of all this super cool stuff we found, we're doing some science, but I always got to pull them back to be like, okay, do a targeted science question. Don't ask everything. Ask what is relevant to what we need to understand right now. And I think that's kind of the joy of what we do and the why we've gotten is gone as far as we have with as quickly as we have with the least amount of money.
Yeah.
We dosed 35 patients already in about 5.5 years. So from the inception of the company, we've already dosed 35 patients.
I mean, that's amazing. When you put it like that, in comparison to where other companies have got. And I think the point you made about being frugal is really, really important, especially, we're in a tough market at the moment where companies are struggling to raise money. And then other companies, well, aren't in some respect, but you still got to be super frugal bit. And even if you can raise, it's like take what you want and then it makes you really efficient with that cash. Just on like the cytokine front, which has obviously been a huge challenge in drug development for years. Why is it been notoriously difficult? And also what makes your approach different?
Yeah, I don't think we've used them in the right way. And I know that sounds kind of crazy, but, to kind of make the immune system work, what we've been doing is, I think, a bit misguided. And so for a proper kind of communication with T cells, we know you need signals 1, 2, and 3. So signal 1 is T cell engaging MHC and antigen. Signal 2 is called co-stim. That's basically both signals into the T cell and further stabilizes what becomes the immune synapse. So you have kind of this affinity play, which is T cell receptor MHC peptide, then an avidity play, which is essentially CD28 B7. And then when you activate the T cells, you then secrete cytokines, and predominantly you're secreting IL-2 and interferon gamma. So that's signal 3. Signal 1 is T cell receptor engagement. Signal 2 is co-stim. Signal 3 is the cytokine. Now, if you think of this as a train, signal 1 is the engine that makes everything start. Signal 2 is the co-stim. Those are kind of the cars in the middle. And signal 3, the cytokines, that's the caboose. So therapeutically, if you try to jack up the huge concentration of the cytokines in the caboose, you're effectively trying to push the immune system forward with the end of the train. So we looked at it a little differently. We asked ourselves, okay, we're taking IL-2. IL-2 signals the same way as co-stim, right? It activates PI3 kinase and RAS and AKT and AP1. That's identical to CD28 signaling. Okay, that's the first thing. Second thing is co-stim augments the affinity and avidity of the T cell for the tumor cell. It makes it stick together better. So what we did with the 2 and the 10, we're delivering 2, which remember is part of signal 3. The 2 and the 10 together synergize to induce interferon gamma. So that's the other part of signal 3. So we've solved signal 3. IL-2, interferon gamma, that's signal 3. But by virtue of the structure and function, we have 2 points of contact of the cytokine receptors on the T cell and 1 point of contact with the targeting vector to the tumor-associated antigen on the surface of the tumor cell. So now we're bridging the gap. Remember, co-stim is an increase in avidity. It helps the T cell to stick to the tumor cell. So with one molecule, we have basically given signal 3 and signal 2. So from that perspective, we then asked ourselves, well, if that's true, the hypothesis is we should profoundly synergize with T-cell engagers. That's signal 1. And the answer is we do. So we proved in our clinical trial that we can give high-dose IL-2, and we see mixed responses. We see some tumor shrinking. We see immune activation, all the good stuff. Now, the next step, the real kind of leap in therapeutic capacity is combining with the T-cell engagers. Now, as it turns out, there's another component of luck here. As it turns out, pharma collectively has made billion-dollar bets on T-cell engagers. And the T-cell engagers have two problems. They are essentially at the dose that they can provide safely, not sufficiently effective. Okay, so what is the safe dose? What is the problem of safety? Well, it's CRS, cytokine release syndrome. What we have proven is that cytokine release syndrome is actually initiated by interleukin-2. We have then proven when we combine interleukin-2 with interleukin-10, we block CRS. We've done that in our patients. We've also shown it in combination with T-cell engagers that we basically are telling the T-cell, you're making enough IL-2, don't make it. And the CRS signal actually comes from monocytes. They're the highest IL-10 receptor density cell type. So the IL-10 shuts down the monocyte, responsive CRS. We tell the T-cell you have plenty of IL-2, and the synergy of inducing interferon gives that whole circuit. So what we then figured out, and there are times when you feel super stupid, and this is one of those times. Well, where I feel super stupid. This is one of those times. So I had assumed incorrectly that I should target the TCE to one tumor-associated antigen and the Diakine to another one, because if I target it to the same one, there might be competition. Well, as it turns out, when you target both modalities to the same tumor-associated antigen, you essentially pre-cluster and you synthetically build the synapse within minutes. So, that's just further our understanding of what nature actually does and how to synthetically build. So what we see is that when you prime T cells with a Diakine with 210, and then you add in a T cell engager and tumor cells to this, when the Diakine and the T cell engager are directed to the same tumor associated antigen, you build the synapse within minutes, and within an hour, 70% of the tumor cells have been destroyed. So the speed of recapitulating, talking to the immune system in the way that is the most efficient and maybe even more efficient than the endogenous system really then drives that therapeutic modality. And again, that IL-10 is present to take away the CRS tox. So we actually think, we have kind of the next evolutionary leap in therapy for cancer patients. That's why we think we're different. It's really the targeting of, or the coupling of the two together and then targeting to the tumor cell surface.
Well, that's one way to be different. A pretty good way to be different.
We think we can cure cancer. That's how different we are.
I just want to this is a question I ask all my guests, John, at the end of the podcast. And normally people, just so people know, normally people need a bit of time to think about it. Not John. He knew his answer straight away. So if you could have dinner with any scientist or innovator, past or present, who would it be and why?
Yeah, it's Nikola Tesla. And, I've read probably every book that exists on his life. And I understand that at the end of his life, he was a little loopy, but I can understand why. And what I mean by that is he had such a profound intellect. And currently, scientists are thinking that maybe our brains are actually quantum waveforms. And if you apply that to his Nikola Tesla's self-described visions of, he would see in his mind when he was sleeping, they perfectly formed whatever it was that he was working on, right? And so he would visualize it before he even built it. This is very akin to Einstein thinking of a thought experiment. Like, think through it, imagine it first before you commit anything, any pen to paper, any building materials to build whatever it is you're looking at. And the reason I would love to have that dinner with Tesla is I think and envision molecules in 3 dimensions. And I'm super curious because I'm always trying to get better at me, at what I do. I'm wondering if he had techniques, if he had kind of, prior to laying down and dreaming these things, if he had a whole thing that he went through that made him so efficient. He's one of the most prolific inventors in the history of the human species. Like, he was doing things that were so profound and so far ahead of anyone else that he literally no one could understand him. And I'm not there. Don't get me wrong. Like, people more or less understand the babble that's coming out of my mouth. But the point being that I would love to just listen to him speak about himself. I think that would be really just a truly profound experience.
I think that's an incredible answer. I think a really good one. And yeah, even better that you knew the answer to that one on the spot. So yeah.
That's not the first time I've thought of it.
Yeah, that's good. That's good. So look, thanks so much for coming on, John. It's been an absolute pleasure.
Thanks, Max. I appreciate it. Really, really appreciate the platform, and I think you're doing a great job.
Amazing. Thank you. And please, everyone, like and subscribe to the BioHub. Thanks again, John.
All right. Cheers.